Hello everyone, I'm Lalarukh an ample scholar from Pakistan. Today I would like to discuss on the topic targeting expression of inflammatory markers in cortex and hippocampus of epileptic mice model treated with novel drug combinations. What is epilepsy? Epilepsy is basically a neurological disorder due to the imbalance of neuronal activity that leads to seizures that could be either caused by genetic or environmental factors. The epilepsy is of three types: generalized epilepsy, focal, and combined generalized and focal epilepsy. It could be either treated with pharmacological and non-pharmacological treatments that includes anti-epileptic drugs, surgery, ketogenic diet, or EEG. According to the study conducted by IA Khatri, it says that approximately 1% of Pakistani population is affected by epilepsy. What connects inflammation and epilepsy? Let's talk about that. Due to hypoxia, trauma, or inflammation, inflammatory cytokines are released. The target cytokines of my studies are pro-inflammatory cytokines, IL-1 beta and IL-6. Interleukin-1 beta acts on a receptor initiating the NF-kappa B pathway that causes the production of prostaglandins and nitric oxide, whereas interleukin-6 acts on GABA and decreases its transmission. Both of these acts on the NMDA receptor, increasing the calcium influx, causing hyperexcitability and neuronal network changes that causes epilepsy. Why did I choose this topic? It's so because about 35 to 40% of the patients are resistant to the available anti-epileptic drugs. Therefore, there's a need to establish new drug combinations that could target different factors. The study includes two objectives. The first one is to evaluate the effect of diclofenac sodium in combination with levetiracetam on epileptic seizures in pilocarpine-induced epilepsy mice models. Number two, to analyze the effect of diclofenac sodium and levetiracetam on expression of inflammatory markers in hippocampus and cortex of epileptic mice models. Methodology. Phase 1 included the animal studies. There were 5 experimental groups that included NMRI mice. All of them were male, around 24 to 30 grams, weighing around 24 to 30 grams. Group 1 was the control group. Group 2 was the combination-only group. Group 3 was the disease model. Group 4 was the standard treatment group, and Group 5 was the combination treatment group. All of these were given the drugs for consecutively 3 days, except for group number 3. On the very third day, all these groups were given pilocarpine challenge, except for group number 2 and group number 1. After that administration, they were observed for 90 minutes for behavioral evaluations. The parameters included the latency to first seizure, latency to continuous seizures, duration of continuous seizures, and percentage survival. After this evaluation, the animals were then anesthetized and dissected. The brain was isolated, and the cortex and hippocampus tissues were collected. The RNA extraction was done, followed by RT-PCR and then cytometric analysis. The results were then run on SPSS to determine the statistical analysis. Results. In phase 1 results, when the survival rate was measured, it showed that there was significant increase in the survival rate in the group of animals in the combination treatment group when compared to the disease model group. The latencies to the first seizure and continuous seizures were significantly increased when compared to the disease model group. The duration of continuous seizures in the pilocarpine disease model group was significantly quite long, whereas the levetiracetam, diclofenac, and pilocarpine group had significantly decreased durations in the continuous seizures. When talking about the molecular study results, the densitometric analysis were normalized using the beta-actin gene. When IL-1 beta was ran on gel, it showed a band size of 357 base pairs and the SPSS results showed that interleukin 1 beta levels were significantly increased in the standard treatment group, whereas the levels were significantly decreased in the combination treatment group. When talking about IL-6 expression levels, when ran on Gen, it showed a band size of 475 base pairs. Spaces showed that there was minimal to no expression levels of IL-6 in the combination treatment group, whereas the standard treatment group showed expression levels of IL-6. In accordance to my study results, it showed that diclofenac sodium increases the efficacy of levetiracetam. This combination decreases the epileptic seizures activity along with the pro-inflammatory cytokines that play a vital role in epileptogenesis. Therefore, after further experimentation, it could be given to epileptic patients. On mice models, it could be used in different dosages to determine the minimum effective dose, and it could be also replicated on chronic model of epilepsy. I would like to thank my mentor and my supervisor, Dr. Farina Hanif, my co-supervisor, Dr. Saima Mahmood, Ms. Kurtalan Armer, my late lab partner, Dania Nasim. I would further thanks Dow Research Institute of Biotechnology and Biomedical Sciences and Higher Education Commission Pakistan for the funding. These are the references. Thank you so much.